Good morning and welcome to our session. Today we'll be discussing the most probable questions to be asked from the chapter Biotechnology 2 Applications. So the type of questions as usual, as well as the thrust points, the most important points from the chapter will be discussed now. Okay? So chapter Biotechnology 2 Applications. Chapter number 12 of second year NCERT. Clear? We go directly to the question. The source of complementary RNA. See, this is a question regarding RNAi. We know that RNAi is a intracellular defense mechanism that has been described towards the end of and the beginning of this, towards the end of the last century and in the beginning of the present century. It was discovered in and worked out in a soil living nematode. What is this RNAi? It is also called the gene silencing. Simply, it is called gene silencing. Even though the gene is present, the gene is not allowed to express because its mRNA will be suppressed, prevented from expression. That mechanism was actually described in a nematode Caenorhabditis elegans. In a soil nematode by name Caenorhabditis elegans. By two eminent workers, Andrew Fair and Craig C. Mello. And this technique we applied in making plants resistant to nematode attack. We have studied that the tobacco plants, which are all attacked by Meloidium incognitia, that will be causing severe damage to the crop, that was cured by this novel technology. Also, we use this technique for producing new varieties of tomato called flavor saver variety of tomato. Okay, what is this mechanism? Normally in eukaryotes, we thought that there is only intercellular defense mechanism. We have studied that in our body we are having a very developed and advanced immunological system that protect us from pathogens, diseases, etc. And that is intercellular. And we also studied that there is intracellular defense mechanism in prokaryotes like bacteria, that is restriction endonuclease methylase system, or we call it as methylating restricting enzyme complex. That is intracellular defense mechanism in prokaryotes. We believe that only in prokaryotes there is intracellular defense mechanism, that is within the cell. There is a defense mechanism. But we came to know the existence of this mechanism, that is gene silencing or RNAi, RNA interference, which is an intracellular defense mechanism. This will be starting its work once there is a double-stranded RNA produced. What is this double-stranded RNA? This double-stranded RNA is not at all produced in our cells. We will be having only single-stranded RNA and double-stranded DNA. And double-stranded RNA is not a normal constituent in our cells. But in rare occasions, you can have a double-stranded RNA. You are having two complementary strands, which will be, these are all two single-stranded RNA that are complementary, so that they will be forming a pair and will be thus forming a double-stranded RNA. Okay, what are the conditions? The formation of a double-stranded RNA is on rare conditions, and that is the beginning of the initiation of RNAi system. Once a double-stranded RNA is produced, which is an abnormal constituent, a special protein called dicer. You have a dicer. What is a dicer? Dicer is a RNase enzyme. It was Emily Bemster who discovered this enzyme and named as dicer. This will be identifying double-stranded RNA only and will be cutting that double-stranded RNA into small pieces called siRNA and miRNAs. Si and miRNAs are produced, which are all immediately given to another protein called risk protein and will be resulting in the formation of risk complex that you have studied. Now, the question here is, under what conditions a double-stranded RNA will be produced? Normally, I told you there will be no double-stranded RNA, only single-stranded RNA will be produced. Here, this double-stranded RNA, an abnormal constituent, will be produced by an infection by a virus having RNA genome. Not all RNA genome, but which is having a retro. It is a retrovirus. Remember, retroviruses are having RNA. All retroviruses are RNA viruses, but all RNA viruses are not retroviruses. But here, having RNA genome, that may be a retrovirus. If you are having an infection by a retrovirus, definitely from RNA by reverse transcriptase. From RNA, a DNA will be produced. First, it will be producing a complementary DNA, cDNA. And that cDNA will be complementary to the RNA, thus they will produce a double-stranded one. Mobile genetic elements. There are very rare pieces of DNA, genome pieces, which are capable of changing their position. They can be called as transposable elements or transposomes or jumping genes. That is why they are called nomadic genes. They go on changing their position occasionally. It is very rare. They have been discovered long years back, as early as in 1940 by Barbara McClintock in Siamese, but we didn't give any importance to her discovery. And later on in 1980s, again the same thing was discovered to be present in other organisms, including our Drosophila. Then we came to understand the significance of her invention, and only then she was awarded the Nobel Prize for her wonderful discovery. Transposomes, nomadic genes. Okay, so mobile genetic elements which are going on changing their position, which is called transposomes, and DNA sequences are also there that will be replicating via an RNA intermediate. There are very rare DNA sequences that require an RNA as an intermediate agent for its replication. So in these conditions, you will be having a double-stranded RNA produced. Is that clear? And whenever you are having a double-stranded RNA, you will be having the RNA activated. It's a very wonderful question, right? Yes. And no details are given in our books, but you should study it clearly. The RNA pathway is initiated by the enzyme. What is that enzyme? It is Thrissur. Thrissur is an RNase enzyme of type 3. It is type 3 RNase, which will be cutting only double-stranded RNAs. Both strands are cut at the same level and will be cut into small pieces of average 20 to 25 nitrile base length. So we take the average as 23, which is having an average size of 23 base pairs. So Emily Bimster, who discovered and named them as Thrissur, which cleaves double-stranded RNA to short double-stranded fragments of 20 to 25, average 23 pair, which is called siRNA, small interference RNA, okay, or miRNA, micro RNA, okay. Emily Bimster discovered them, who will be cutting the double-stranded RNA into small pieces called siRNA or miRNA. The Thrissur will be cutting both strands. It will be cutting only double-stranded RNA, not single-stranded RNA. Okay. Steps involved in the production of humulin. What is humulin? The first human protein that was produced by rDNA technology by transferring human gene to E. coli. See, we were erstwhile giving insulin taken from other animals. That is not such effective and sometimes it may be evoking some allergic responses. So we thought of giving the gene of human beings for insulin to microbes and producing the insulin. But we are having so much of hurdles, so much of problems in that. The main problem is that we cannot get fully functional, mature insulin, because the insulin produced will be always in an immature form, pro-insulin, and there is no editing machinery and enzymes needed for editing and making the pro-insulin converted into, there is a pro-insulin converting enzyme needed for converting the immature pro-insulin to mature insulin. Prokaryotes are very poor in their machinery, in their mechanism. So that was a problem. So we had a technique. That technique was developed by an American company, multinational company, Eli Lilly. Okay. And what was the technique? The technique of the production of human insulin, which they called as humulin, is this one. They will be producing synthetically the chain for alpha and another chain for beta, polypeptide chains. So these are the artificially synthesized DNA. One response for alpha chain, one response for beta chain. Okay, and they were all given to two different E. coli. Another E. coli. And both of them were cultured in two separate bioreactors so that they will be producing alpha protein and beta protein, or alpha chain and beta chain. They were all extracted, purified by downstreaming process, and they were all mixed together under certain conditions, during which suddenly there will be the formation of disulfide bond between alpha chain and beta chain, and thus functional human insulin, which is the perfect insulin, ideal for human, was produced. That insulin is called humulin. So what is the procedure? First one, See, there's a small problem in the numbering. There are 2 ones. That's why I have cut it and written like that. Synthesis of gene for human insulin artificially. We are having a technique of producing any gene artificially that will be coding for a particular protein. Then we have to insert the human insulin into the plasmid and give them to the 2 strains of E. coli. Okay, so introduction of this recombinant plasmid into E. coli. And what will happen? They will be cultured in the recombinant E. coli in bioreactors and extraction of the recombinant gene product from E. coli. And finally, we are getting the finished product, purification of humulin. These are the steps that were followed by the company, multinational company, American company by name Eli Lilly and Company in 1983. Okay. So the order should be this one. 1, then it should be 4. So 1, 4. Immediately we came to the answer. Okay. Gene therapy. What is gene therapy? It is yet another application of our genetic engineering for replacing a defective gene with a healthy one. Human beings are all suffering from a large number of genetic diseases, mostly because of gene mutations, and we are trying to have a remedy for such diseases. And the first successful gene therapy history comes from the treatment of Ashanti De Silva, a 4-year-old girl. In 1990, September 14, she was given a treatment at National Institute of Health, Bethesda, by Dr. French and Dayson. Okay, what was the procedure? The procedure, she was suffering from severe combined immunodeficiency disease. We simply call it as SID. So, we took a healthy gene, normal gene for ADA. See, it is because of the deficiency of this enzyme, adenosine deaminase, that she was suffering from, and other people were suffering from the disease, severe combined immunodeficiency. It is a immunodeficiency disease because of the mutation of the gene, adenosine deaminase gene. This adenosine deaminase is essential for rapid multiplication of cells, for the production of blood cells. Without this gene, no enzyme is produced, and without that enzyme, no cell multiplication, rapid multiplication is occurring. That means the lymphocytes, immunologically competent cells are not at all produced. So that patient will be having defective and deficiency in the immunity. Okay. And what was the technique? Correcting of gene defect that has been diagnosed in a child or embryo. That is the main purpose of gene therapy. We are not treating, we are all replacing the defective gene with a healthy one. Correction of a genetical defect involved in delivery of a normal gene into the individual, correct? You are correcting. Delivery of a normal gene into the embryo or to a fetus to take over the function of and compensate for non-functional gene. Okay, that non-functional gene is replaced by a healthy gene. A corrective therapy for a hereditary disease. We believe that we may be in future capable of correcting all such genetic diseases by this technique. Okay, treating of an infectious disease with immunopotential. No, gene therapy is not treating of infectious diseases with immunology. That is called immunotherapy. This procedure is called immunotherapy that we are all giving to cancer patients, etc., etc. Not we are not treating the patient with defective gene with a corrected or normal healthy gene. The technique that do not serve the purpose of early diagnosis. What is early diagnosis? Normally, we treat a patient only when the symptoms are expressed. We'll go to a doctor and consult, and he will diagnose by analyzing our blood, sputum, or urine, as the case may be, or you may be having scanning done. These are all diagnostic techniques. When the symptoms are already expressed, now you try to analyze what actually is the reason, what is the disease, etc. by this mechanism. Now, in early diagnosis, we are trying to analyze whether you can have a disease long before the symptoms appear, so that the treatment will be very simple and completely cured. You can cure the disease completely because you started the treatment even when the symptoms have not started, the pathogen had not become very virulent. Okay, so you will be having a chance of complete curing of that, cured from that disease. Now, the technique that do not serve that purpose of early diagnosis. Normally we use the three techniques: PCR, whereby you can detect whether you are having an infection by a bacteria or a virus, because even a very small dosage of number of cells of bacteria or small viral molecules can be amplified. They get amplified by PCR, and you understand that, okay, you are already having a mild infection, the pathogen had entered you. Those symptoms have not appeared, you can start treatment for that. Then, whether your genes are normal or mutated, this can be diagnosed by taking your cell, taking the DNA and performing a autoradiography. By treating with a probe, which is a normal gene. Or you can also have ELISA test done. It's a highly sensitive antigen-antibody reaction, whether your body is having an antigen or your body has started producing antibody. This can be detected by ELISA technique. These are all commonly used, okay, for early diagnosis. So, recombinant DNA technology. So, we are all using that recombinant DNA technology so that you will be having production of large number of cloning and analyzing them. Okay. Polymerase chain reaction, PCR, enzyme-linked immunosorbent assay, ELISA, DNA hybridization, which is used in autoradiography. Okay, so these are all techniques. Whereas serum analysis, you are taking your blood and analyzing for abnormalities in it. Okay, that is a technique that is currently followed when you are already showing symptoms for that sickness. It is not an early diagnosis. All others are early diagnosis, whereas serum analysis is not an early diagnosis. It is a traditional diagnostic method. VTTR, variable number tandem repeats. What are they? Remember, in our DNA, only very small part of our DNA will be coding for proteins. Most majority of our DNA are not coding for any protein. Okay, and we are all separating our DNA based upon their density gradients. The heavy DNA that is separated first will be called the core DNA. They are all heavy DNA. We will be having a more concentration of CG pair. They are all very heavy. Whereas the other DNAs, which are lighter, which will be precipitated and separated in density gradient centrifugation, they will be called satellite DNA. These satellite DNA are mainly composed of a special type of DNA which is called repeating tandem units. That satellite DNA is mostly made up of small length of DNA in which nitrogen based sequences are coming repeatedly. That is what is called tandem repeats. These tandem repeats are showing maximum differences. Differences at the molecular level of DNA is called polymorphism. So we say that they show maximum polymorphism. Who is showing that maximum polymorphism? Variable number tandem repeats. They are not coding for any protein, and so they will be inherited from generation to generation. So they show maximum differences. They will be different in different persons. Okay, and even among identical twins, there may be slight differences at that type of DNA. That's why we take this VTTR as a criterion for recognizing, identifying the nature of the DNA of an individual. That unique features of the DNA of a person is called DNA fingerprints. Just like the unique nature of our fingerprints, the DNA is also having some unique features which can be easily identified in the VTTR segments. So what is that VTTR? VTTR is new terminal region of DNA, short sequences of proteins, specific non-coding. That's very good. It is not at all coding for any particular type of DNA. They constitute major part with unique tandem repeats. See, say for example, this is a tandem repeat. I will show you what is a tandem repeat. Easy understanding. AC, GC. Again, AC, GC. AC, GC. AC, GC. Like that. The same repeats of nitrogen based sequences are coming for thousands and thousands of times repeatedly. Same sequences are repeated, and the repeats are in the same order. That's why it's called a tandem repeats. What's the meaning of tandem? In the same order. So ACGC, ACGC, they are all very close to the original one and are in the same order, coming in very large number of repeating units. Okay, that is why they are called tandem repeats. And these variable number tandem repeats are the basic for the unique nature of DNA of one person. Okay, they are unique tandem repeats. Consider the following statement regarding DNA sequencing and arrange them in the correct order. How you are all doing that DNA sequencing? So you may think that these are all part of chapter number 6, molecular biology. Okay, but here we teach the DNA sequencing, human DNA fingerprints, etc., along with our application of biotechnology. Because they are all used. They are all actually used for analytic purposes in which we are using the biotechnology principles. That is why we are all using it. So, what are the steps in DNA sequencing? We are having the DNA of that individual taken from a cell, so DNA isolation, that is the first one. Okay, after that, we have to digest them with the restricted endonucleases so that they will be cut into so many pieces and then they are to be separated. Resolved, that is called analysis of restricted fragments by age. Then we have to give it to bark and yak. bacterial artificial chromosome and yeast artificial chromosome for cloning. We cannot use plasmids because it is eukaryotic genome. Eukaryotic gene cannot be normally carried and multiplied in a prokaryote. That's a problem. Okay. So we have to use artificial chromosomes which are bacterial and yeast and given to the eukaryotes like yeast or mammalian cells, etc. They were all used as the back and yak, not wa. It should be yeast artificial chromosomes, back and yeast artificial chromosomes. Then what should be done? Fragments are sequenced using automated sequencers. These are converted first into single-stranded DNA, and after converting into single-stranded DNA, they are all to be given to automated sequencing machine, which work on the principle developed by Frederick Sanger, the great man, who also developed a technique for sequencing amino acids in a protein in 1950s, for which again he was given the first Nobel Prize, and now in 1980s he was given Nobel Prize for developing a technique. What was the technique? It is called dideoxyribose. Technique or chain termination technique. That is the technique he used for identifying the nitrogen base sequences in a segment of DNA or RNA. Okay. He developed that technique, and the machine that we used is an automated computerized machine which will automatically sequence and tell you what are the nitrogen base sequences in that piece of DNA. Actually, that machine was developed by Lerog Hood of the University of Seattle, not University of Washington, in Seattle. Lerog Hood. That is the automated machine. Okay. He developed. So we give this single-stranded RNA or DNA to that machine. Now the machine will tell you what are the nitrogen base sequences. Now we have to align them, properly align the pieces. For that alignment, it's a very complicated procedure. We have to make the same DNA cut by two different restricted enzymes so that they will be having some overlapping sequences. It will take more time for explaining what these overlapping sequences is that. You have studied all these things in the classes, in the theory classes. So we have to use two different restricted enzymes for cutting the same DNA, and these are all given for sequencing to the computer. Then the computerized machine will be telling you that there are some overlapping machines, and you have to make them align properly. Based upon these overlapping sequences, you have to align these pieces properly, and then only, so for example, this may be the piece number 1, this may be piece number 7, this may be piece number 4, this may be piece number 10, like that. They have to be aligned properly. Then only finally you will be getting the full picture of the nitrogen base sequences of the entire DNA of that chromosome. This is what is called alignment. Okay. So there are two steps. First is identifying the nitrogen base sequences in pieces of DNA. 2. Proper alignment. For this, the computer will be helping you. The second stage is more complicated. We have to make physical map, genomic map. We have to mark which piece of DNA are all forming a particular gene, where it is the location, etc. What is its sites, what are the nitrogen bases, etc. That is what is called gene mapping. That is not with the help of computer. It is with the help of certain other mechanisms like the satellite DNA and restriction enzyme polymorphism mechanisms. It is an entirely different one, not assisted by computers. Okay. So finally, you have to arrange these specimens based upon the overlapping sequences. That is how we are all aligning. Alignment. What is alignment? Join the pieces in the proper way so that you will be getting the continuous sequence of the entire DNA of that chromosome. Clear? Methodology of sequencing, which involve identification of all genes that are expressed. See, in sequencing our DNA, we are having two techniques. The first technique is ETS, expressed sequence tagging. See, our DNA of a chromosome is a very long one from the molecular level. It is a very long one. So sequencing is not that easy. So what we did early is that we took small piece of nucleic acids. What are the small piece of nucleic acids? We know that a DNA will be a double-stranded one and it's a very long one, whereas the RNA it produces will be a very small piece and a single-stranded one. So reading a single-stranded small piece is easy than reading a complicated long double-stranded DNA. So we took these small pieces. What is this small piece of RNA that are coming from DNA? It is mRNA. So we took that mRNA, which are all produced by a cell, and give it to sequencing. So those which are all expressed as mRNA, that is what is called express sequence tagging. That was the technique that we used in the beginning in Human Genome Project. But there was a problem. What is the problem? The technique is easy, but only 2% of the DNA will be coding for mRNA. We understood that only a small part of the DNA will be coding for RNA. And suppose we are all taking only that coded part, exon part, and identifying the sequence, we get information only on 2%. That means we are not aware of the remaining 98% which is not at all coded. So we are not in a position to understand what is the sequence of the entire DNA. So we abandoned this technique and we went to the second technique called sequence annotation. This is called sequence annotation. What is sequence annotation? First, we will be identifying the sequences and then we will be annotating. That is what I told you. We have to identify which part of that long DNA is constituting a gene, what is its location, what is its size, and what are the other genes, etc. in that sequence. That is what is called annotation. We are all annotating after sequencing. Clear? So it's called sequence annotation. So this technique is sequencing which involves identification of all the genes that are expressed as RNA. Don't get confused. All the genes. That is just to confuse you. They are all expressed as RNA. Who will be expressed RNA? That is called expressed sequence tagging. in that technique, we are not aware of, or we are not getting information on those which are not at all expressing. Okay, those which are not expressing. Only genes which are all expressed, that is producing mRNA. That alone will be sequencing, because we are taking that mRNA and giving for sequencing. So that is the greatest defect in EST. What is that? It forms the base of DNA fingerprinting. See, DNA fingerprinting, we are all finding out the unique nature of the DNA of every individual. And where is our uniqueness? Remember that 99.9% of the DNA of different persons are identical. In Human Genome Project, we identified that in 99.9% there is uniqueness. That means we are having differences only in a very small part of the DNA. Okay, so we have to look into that part which is easy to identify a person. We have to look into that part of the DNA which shows maximum differences. That difference is in the tandem repeats. So, what is that form the base of DNA? DNA is different only in tandem repeat part, which is constituting our major part of the DNA, and tandem repeats are all highly different. They are all showing maximum polymorphism, and that forms the base of our identification. So, where is the tandem repeats? Tandem repeats are the DNA that are mostly constituting the satellite DNA. Satellite DNA occurring in highly repeated short DNA segments. These short DNA segments are called the variable number tandem repeats. Okay. So it is in the satellite DNA. That satellite DNA is mainly made up of variable number tandem repeats, meaning made up of same repeating units of DNA. That is the basis of our DNA fingerprinting. What is Thrissur blotting? Blotting techniques are a technique for transferring these biomolecules from the separating medium to a stronger medium. For example, we have studied that our DNA is separated in a agarose gel. Okay. We are having it in a agarose gel. It's a glass plate in which we are all preparing a particular concentrated solution of agarose and pouring the agarose solution on it, and that acts as the medium, and the DNA will be resolved, separated as orange colored bands. We can see, visualize after resolving. Now, the DNA is in that agarose plate which is on a sheet. It is a very powdery one. Okay, so will be easily be damaged. It cannot be transferred. It cannot be used for other purposes. And we can, of course, isolate each chip, each piece, and transfer to a suitable solvent. And S.M. Southern developed a technique whereby he transferred these DNA pieces on the agarose gel to a strong paper like nitrocellulose or nylon. Or even in a filter paper, so that you can use it for other purposes. Okay. This is blotted. What is blotting? When there is extra ink falling on your paper when you are writing with ink pen, what you will do? You will use some substratum like chalk piece which will absorb that extra liquid. That is what is called blotting. When extra ink is flowing, it will be absorbed, okay, by a blotting paper. What is the peculiarity of chalk or blotting paper? They are having small microscopic pores, small capillaries in them, which will be absorbing the liquid very quickly into them. That is why we are using a chalk piece and not a rubber or an eraser. Okay, so we use such a paper, just like a filter paper or nylon paper or nitrocellulose paper. You keep it on this agarose gel and keep it in a suitable medium. Okay, so you are having a suitable solvent that will be dissolving the DNA. You keep a large amount of, say for example, filter papers and keep a heavy weight, just like a paper weight, okay, over it, so that it remain pressed. So what will happen? Gradually by capillarity, this fluid or solvent enter into the agarose gel and it get gradually migrated to the DNA pieces, and the DNA will be immediately dissolving in it. Once it is dissolved, that liquid is immediately blotted away. That fluid is now blotted away. In a corresponding position, the DNA will be absorbed, blotted by the nitrocellulose paper, and the fluid part will be blotted away or absorbed by the filter paper that you are all keeping above it. That's why we are all using the filter paper, common filter paper. This is the common filter paper and this is the glass weight that we are all using. This simple technique was developed by S.M. Southern and is called Southern blotting. Okay, for blotting of DNA. Later on, we developed a technique for blotting away RNA molecules that was called northern blotting. And we have studied that the confirmatory test for AIDS is western blotting. So, southern blotting, northern blotting. Southern blotting for DNA, northern blotting for RNA. Western blotting is for transferring protein, which are all separated in polyacrylamide gel electrophoresis. Polyacrylamide gel electrophoresis for protein. That is the confirmatory test for AIDS, northern blotting for separating proteins. Now you understand what is blotting technique? Okay, now you go to the question. No details are in our books. That's why we have taken such questions for you deliberately. You should understand the techniques. Southern blotting refers to transfer of DNA fragment from nitrocellulose membrane. No, it is in the reverse. Electrophoretic gel to nitrocellulose. Transfer of RNA, no. Transfer of DNA fragment from electrophoretic gel. Okay. That is agarose gel into nitrocellulose membrane. Correct. Transfer of RNA fragment from electrophoretic gel into the nitrocellulose membrane is called northern blotting. Is that clear? Which of the following is associated with the flavor saver tomato? I was telling you, flavor saver tomato is a new variety of tomato that have produced by RNA interference technique. We are suppressing the expression of a gene, polygalactouronase enzyme, that is responsible for bringing about changes in the skin when the fruit is ripening. That is responsible for easy damage of the tomato. So, we are preventing such changes with suppression of the gene responsible for the production of that enzyme, which is responsible for such changes. Encourage eco-friendly herbicides? No. Reduce herbicides? No. Eliminate weeds? No. Eliminate weeds from field? No. Reduction in post-harvest. See, tomato will be harvested in a particular season and the whole market will be full of this tomato and they have to be sold out farmers cannot store them because they will be easily be perished. So after harvest, the farmers are forced to sell them at whatever small price they will be fetching. So they will be incurring a lot of loss. So there is post-harvest damage. After harvesting, the fruits are easily be damaged. They are having no shelf life at all. Actually, they will be damaged within 1 week of or 10 days of harvesting because they are having very poor shelf life, storage life. We have to increase that shelf life. That is, we are all making them to have no loss. Even after harvest, you can keep it for a year, no problem. Reduction of post-harvest losses. Advantage of genetically modified crop include, see, why we are all modifying plants? We are modifying plants in such a way that they become highly tolerant to unfavorable abiotic conditions, like they may be tolerant to drought or extreme rain, decreased post-harvest losses, just I told you, just like the tomato, increased reliance on chemicals. No, we want the plant themselves resistant to the pest so that we need not add any pesticide. See, adding pesticides will be having two effects. One, we are using chemicals that is ultimately harmful to the consumers itself. Two, pesticides are very costly, so the farmers are incurring huge expenses in agriculture. We have to reduce the input of agriculture. Okay, then only agriculture become profitable. So we have to reduce the reliance on chemical pesticides. We need not depend upon chemical pesticides. We want to make the plant itself resistant to pest. That's why we have made BT varieties. Okay, increased efficiency of mineral usage. They will be judiciously using very small amount of nutrients that they absorb from the soil, so that soil is not depleted of its fertility and we need not add fertilizers very frequently. That also will be reducing the dependence on agro-based chemicals. So pesticides and fertilizers are agro-based chemicals and we have to reduce the dependence on them. Okay, because both are pesticides as well as fertilizers are chemicals that are ultimately harmful. Okay, they will be even damaging our environment. And reduced post-harvest losses and enhanced nutritional value. See, post-harvest losses, typical example I told you, flavor saver, and nutritional value, that is the vitamin A enriched yellow colored rice, golden rice. Okay. We are enriching, increasing the nutritive value of these products. Now, what are the disadvantages of insulin that are, so till 1983, we were all using insulin that is extracted from other animals. How we will be taking, they will be taken from slaughterhouses, from pigs, etc. And when we are all taking the insulin from such animals, what will be? They are all taken from another species. When it is introduced, it will be having less effect. Two, when a foreign protein is introduced into your body, that will be sometimes evoking allergic responses. So these are the two main problems. It is less effective. It would elicit an immune response. It cannot act inside humans. No. It may be acting and it will get denatured. No. So the first two, A and B. That's why we are trying for getting a human insulin itself. And the first protein, we are now having more than 30 different types of proteins, of which 12 are available in India. Okay, these genetically modified organisms are providing a lot of beneficial proteins. Okay, of which 12 are now available in India. The first one is human insulin. A very low concentration of bacteria or virus can be detected. This is not a diagram indented here. Okay, just correct. So you simply forget this diagram. How you can detect very low concentration of bacteria or virus? Suppose your body had been infected by a very small amount of bacteria or virus. You perform a PCR because the PCR will be amplifying the gene, they amplifying the DNA if the DNA of that bacterium or virus is present in your body. Okay, so if amplification occurs, it is positive. You are positive for that pathogen. Okay, so ELISA is antigen antibody sensitive test. Molecular probes are used for detecting whether your DNA is a normal one, healthy one, or a mutated one. Western blot and southern blots are not diagnostic technique. They are all used in diagnosis. Okay, a technique used in diagnosis, but not as such a diagnostic technique. Okay, so PCR amplification gene is multiplied to millions and millions of times, and that is an easy technique of identifying whether one bacteria or one virus is already there. Because from one, you can get large number of copies by that technique. Now, genetically modified has been used to create tailor-made plants. What is tailor-made plants? Plants that are all made as per your desire, that will fit your requirement. That is what is called tailor-made. Okay, to supply alternate source to industries in the form of starches, fuel, and pharmaceuticals. Agricultural products are used in getting pharmaceuticals, fuels, or starches. So we should have genetically modified plant that will be yielding more starch, more concentration of pharmaceutical chemicals or biofuels. We are now using fuels, biodiesel. Okay. Over 95% of all existing transgenic animals are mice. Why we are making more mice which are genetically modified than any other animal? Why we are not producing other primates? Because our genes are 90% closest to mice than any other animal. Okay, we may think that our genes are very close to other primates. No, it is more related to mice. That's why. And basmati rice is distinct for its aroma and flavor, and about 27 documented varieties are. This is a clear case of bioparasite, kasmati, texmati, yes. Most of the industrialized nations are rich financially, and biodiversity, no, they are very poor in biodiversity. They are all very rich in money as well as in technology. Whereas the third world countries are very rich in biodiversity and inherited knowledge. We are all having great traditional knowledge, and intellectual property right that is misused by these Western countries. Intellectual property of these third world poor countries are pirated, like biopiracy. So this is wrong. So 1, 2 correct, 3, 4 wrong. So the option goes to doll. What is that forms the base of DNA fingerprints? The DNA fingerprints are based upon satellite DNA occurring as highly repeating short DNA segments. The same question we have discussed earlier. So that is a very important one. Okay. The base of DNA fingerprints. DNA fingerprints are made upon the tandem repeats, short repeats. Two individuals showing DNA polymorphism, that is differences at the molecular level, may not have much variation in their phenotype. That is what I told you. Two identical twins, they are all identical mirror images, but though there are no differences in their phenotype, genotypically there is differences. Why? Because the differences are present in non-coding part, and characters are because of the coding part. More differences at the molecular level, DNA polymorphism is occurring in the non-coding part. In coding part, there will be no much difference. That's why the two individuals are maybe looking alike. They are all twins. But still, at their molecular level, in the non-coding part, there will be differences. Okay. So, because the polymorphic DNA arises as a result of mutation in the repetitive DNA, which is not expressed. That satellite DNA is not coding for any protein. So, it is not available for natural selection and elimination. So, it is transmitted from generation to generation for millions and millions of years. So, these variations are all accumulated in the non-coding part for generations. That is why you are all having more polymorphism, more differences in the non-coding part. Suppose a mutation occur in the coding part, that will be bringing about a change in the character, and whenever a gene mutation occur, the character it control will be mutated, and these mutations are deleterious, harmful to the processor, and natural selection will eliminate. So, for example, within 10 to 11 generations, that character will be completely eliminated from the population. So, mutation in the expressing part will be eliminated and will not be retained for longer time, whereas mutations or changes in the DNA in the non-coding part is not bringing about any change in the character, and so it is not at all eliminated. So it is remaining without any elimination in the non-coding part and is transmitted from generation to generation for thousands and thousands of generations. So polymorphic DNA arises as a result of polymorphic DNA means difference in the DNA is more in that part because of mutations in the repetitive DNA. What is that repetitive DNA? That repetitive DNA is mainly present in the non-coding part, which is not expressed. Not expressed means it is not at all responsible for any character. That gene is not expressed as a character. It is not a exon. It is only a intron. Okay. BT cotton, not BH cotton, it is BT cotton variety that was developed by introduction of toxic gene of Bacillus, BT gene of Bacillus thuringiensis is resistant to, this is the diagram showing the same NCERT book, the cotton bollworm. They become resistant to the pest. So we are all making pest resistant varieties, cotton, soybean, tomato, brinjal, etc. we have produced, and only cotton is allowed to be cultivated in India. Other edible BT varieties are not allowed to be cultivated in India. So insect pest, there are resistant to cotton bollworm that will be eating away and reducing the quantity as well as quality of the cotton wool. Match the following. BT, Bacillus thuringiensis. It is definitely cryproteins. So A is 4 and there is only one option, A is 4. Immediately we came to the answer. Thermus aquaticus, it is the bacteria which are living in hot springs from which we are getting the Taq polymerase which are all used in PCR. So it should be DNA polymerase. Agrobacterium tumefaciens, nature's genetic engineer, which is a natural vector. It is having a plasmid called TA plasmid, which is having a tDNA. That tDNA is transferred to dicot in which they become infected. Okay, it is causing crown gall disease, the pathogen, dangerous pathogen, but now we are all making them as one of our great friends as a genetic engineering vector. And Salmonella typhimurium, that is the relative of E. coli from which we obtain the plasmid and produce the first recombinant DNA. So, construction of the first rDNA molecule. Again, another question. See, these are all the previous NEET examination questions. See, same questions are all, or same parts are all overlapping. That means that part is very, very important. BT cotton always you will be getting question from BT, Bacillus thuringiensis, ADA deficiency, adenosine deaminase. That is the reason for the severe combined immunodeficiency disease that has treated by French Anderson on a 4-year-old girl, Shanti De Silva, a Sri Lankan girl. Gene therapy, successful gene therapy was done. RNAi, this RNAi is intracellular defense mechanism. Craig C. Mello and Andrew Fair. And PCR, Terry Mullis, he developed that technique, the great American. PCR is used for amplification of genes, so that even a very small presence of DNA or virus or a bacterium can be detected. Detection of HIV infection. Okay. Golden rice. Rice which is enriched with vitamin A. We took that gene for yellow carotene, which is the rich source of vitamin A from daffodil plants. Okay. Genetically modified crop plant that were incorporated gene is meant for incorporating vitamin A. Vitamin A, see, rice is very poor in vitamins and is rich only in carbohydrate. So those children who are all having rice as the staple food are deficient very often in vitamin A and will be having very often eye problems. World Health Organization found that children who are all suffering from vision problems are mainly having rice as their staple food, and this is the reason. So they asked the international community to develop a rice variety that is very rich in vitamin A. Thus, the Swiss Federal Institute of Technology has developed the first successful golden rice in which vitamin A is present in the edible part of the rice. That is golden rice, which is very rich in vitamin A. Satellite DNA is important because what is satellite DNA? I told you, lightweight, which is very AT rich. It is having more AT rich, which is mainly present in non-coding part. Shows high degree of polymorphism in population and also the same degree of polymorphism in an individual, which is heritable from parents to children. See, same degree of polymorphism in an individual. What is the meaning of that part? You take any cell, you take a skin cell, a hair cell, blood cell having nucleus, or any cell, amniotic cell, semen, anything. All cells are having the same degree of polymorphism. That's a peculiarity. So, you take any cell from an individual, they will be having the same type of DNA polymorphism, which is heritable from parents to children. That's why we are using this technique in detecting or in establishing disputed parentage problems, in solving that disputed parentage problems. Now, courts are taking them DNA analysis, okay, as a clear evidence in disputed parentages, because they are all inherited from parents to children. Remember, we are just comparing the DNA, not sequencing the entire DNA in DNA fingerprinting. DNA fingerprinting is just comparing whether this DNA is similar to that of that DNA. That's all. So, if a child is having its DNA similar to that of the parents, it will be having. Okay, that's how we are detecting. Say, for example, you are having a disputed man, and this is the child, okay, and the mother. The child will be having two members of a pair of chromosomes, which is inherited from its mother. So one will be from the mother and one will be definitely from that suspected father. And if any of these two member of chromosome is similar to that of this one, then this man is the biological father of that child. That's how we are all established. Okay. Which kind of therapy was given to? That was what I was telling you in 1940, September 14, at National Institute of Health, Bethesda, a 4-year-old child with adenosine deaminase, ADA deficiency. The disease is severe combined immunodeficiency. It is gene therapy. Dr. French Anderson made the surgery. Okay. That was done with the help of 2 sets of retroviruses. One is a vector and the other one is a helper. Okay. So there's a long history behind that. And he became successful in treating, and this treatment was given to so many other children also. And the only thing is that the treated patient had to come for repeated therapy. Why? Because it was to the lymphocytes that the molecular surgery was done, and lymphocytes are not eternal. They will be living only for a short period, after which they will be dead. So new lymphocytes are formed. Again, that new lymphocytes formed will be also having defective gene. So again, that is to be given treatment. So they have to be given repeated treatments. Had the treatment been successfully given to an embryo, to the bone marrow cells, which give rise to the lymphocytes, in one therapy itself, in one sitting itself, the result will be a permanent one, a permanent success story will be coming out if we are able to give the treatment to the fetus erythrocytes or bone marrow cells. Bone marrow cells. Okay. Now, use of bioresources by multinational companies and organizations without authorization. See, the government there should be taken into confidence, and they should be given compensation. Okay. And without any compensation and authorization from the concerned country and its people, then it is called biopiracy. Okay. We are having biodegradation, that is degrading, decomposing of biological waste and other compounds. Biopiracy is the correct word, pirating of living things. Bio-infringement. What is infringement? Violation of a right or a law regarding living things and living molecules. If that violation is done, then it is called bio-infringement. Bio-exploitation. We are exploiting a living thing or a molecule of a living thing. In India, GEEC, Genetic Engineering Approval Committee, is responsible for assessing the safety and introduction of genetically modified organisms. They will be giving sanction for the release of such genetically modified organisms. They will be approving. Only then there will be such genetically modified organisms will be introduced. A new variety of rice was patented by a foreign company. What was that foreign company? Rice Tech Incorporate of Texas. And they marketed a new, which they claim to be a novel variety of basmati rice. It is called Kasmati or Texmati, though such will have been present in India for a long time. This is a clear case of biopiracy by a multinational company. Okay, that is basmati rice. It was actually produced by crossing Indian variety with a semi-dwarf variety of rice. And they claim to be a novel one, but it was not a novel one. It was a hybrid produced by crossing Indian variety of basmati with a semi-dwarf variety, which they named as Kasmati, Texmati, etc. Now, this is what I was telling you. Golden rice. This golden rice was produced by introducing 2 genes to the rice plant. That is, one Psi gene cytine synthetase that was taken from daffodils. Daffodil is a wild plant with very beautiful yellow colored flowers. And CRT1 from a soil bacterium, Erwinia uridovira. Okay, so that golden rice is a new variety of rice which is enriched with vitamin A. Okay, so it is with vitamin A enriched with a gene from daffodil. See, this part is not given in our book, so you may have a doubt. That's why I have given this explanation. It is not easy to get the gene from carrot. You may think that carrot is having this carotene, you get it, but it will not be expressed properly because we want this gene to be expressed in the endosperm, in the edible part. And for that, they used a technique, a very simple technique rather than a complicated procedure, and ultimately became successful. We want to have the vitamin A not in any other part, not in stem, not in leaf, but in the edible part, that is in endosperm. So that is the ideal one. Gene, cytosine synthetase from daffodils. Expressed sequence tags, ESTs, that is what I was telling you. The first method we tried in sequencing our human genome project. We'll be taking all that RNA that are all expressed, that is mRNA coming out from DNA, that is expressed sequences, they are all given for sequencing. So genes expressed as RNA, we are taking that one and giving to or given to the sequencing machine so that they will be sequencing and will be able to identify the nitrogen-based sequences, but only of this exon that will be producing only the RNAs. Express sequence tagging. And what trigger the activation of protoxin? See, the bacterium Bacillus thuringiensis is having the gene. Okay, it is having the protein granules, that is crystals formed. It is not at all harmful to this bacterium. It is harmful only to those which are all eating down. Why it is not toxic to the bacterium itself? Because here it is in the form of a crystal, insoluble protoxin. That protoxin is converted into active toxin in an alkaline pH. That alkaline pH is present in the intestine of insects that will be eating these parts. Okay, that's why in the intestine, the protoxin will be converted to toxin, and that toxin will be causing the epithelium to be damaged. The intestinal epithelium of insects that eat them will be developing small pores through which water enter, cells burst and break the intestine. So intestine is broken because of this action. Okay, so it is the alkaline pH of the gut that will convert the protoxin to toxin. See, protoxin is crystal, not soluble, whereas toxin is soluble. Okay. And with that, we come to the conclusion of the discussion of the chapter. Most important questions from this chapter. Okay. Hope you have enjoyed that, understood the thrust areas from this chapter, understood these details which are not given to our book. So please understand the subject at its base. You should have a basic knowledge on these principles. Okay. And then only you can answer any question concerned with these techniques. And with these words, I conclude my session and will meet with another set of questions from the next chapter. Until then, goodbye, take care. Thank you all. Thank you very much.